Okay, good morning, welcome everybody to our presentation as part of the Gracious project, the EU Horizon 2020 project, where we're going to be talking about the regulatory area around nanomaterials, and in particular the changes to REACH that are specifically looking at nanomaterials. Your presenters today are myself, Neil Hunt from Jordas Group in the UK, and Dr. Hubert Rauscher from the European Commission Joint Research Centre in Ispra, Italy. So a quick introduction that this presentation originally was intended to be an internal project presentation for Gracious members to give them an idea of what's going on with the regulations. However, we got such interest when talking to other people that we thought it'd be a good one to open out to industry as well. And thank you very much to everyone from industry or academia and the Gracious project who have spent giving up your time to come and see this presentation. The reason I'm just saying this is for Gracious members. I will be presenting a very small introduction to Gracious at the end of my presentation and how it fits in with the EU, the REACH regulations, but I'm giving a very simple introduction, and if it does rather simplify a lot of your very, very hard work, I apologize now, but I've tried to do my best and try and link it into what people who are trying to meet the REACH regulations do. So no further ado, we've topped the 100 attendees mark, so I think it's time to get moving. I would now like to introduce Dr. Rauscher to do the first part of the presentation. And Hubert, as I said before, you'll have to go and give me a heads up to go and advance the slides. So I'm going to be quiet now and pass it on to Hubert. Thank you very much.
Thank you very much, Neil. Hello, everyone. Good afternoon, everybody. I'm Hubert Rauscher from the European Commission's Joint Research Centre. I'm glad that there's such a big interest in our webinar. Neil and myself shared the task of presenting. So I will start with the first part, more on the general regulatory framework for nanomaterials and definitions of nanomaterial, and then Neil will go into the more REACH specific part. Before I start, I have to draw your attention to that little disclaimer down on the left side, that this presentation are our views and do not necessarily represent an official position of the European Commission or an EU agency. And with this we jump right into the webinar. Next slide, please. Can you change that?
Just a minute. Yeah. Sorry, can I just have a quick, I forgot to say something. If people have questions, then please type it in the questions box and we'll try and answer a couple of them at the end of the session. Clearly, if we end up with hundreds of questions, we're not going to be able to do that. So our intention then is to see if there are some questions that keep on appearing from multiple people, and then we will go and send up a follow-up email to the attendees to do our best to answer these questions. If it's a one-off question, then we'll do our best to catch up with you. I should point out that although Hubert is in the European Commission, he's not part of any ECHA help desk or Caracal, and neither am I. So they're not official regulatory answers, they're just our best best views. So if you want an absolutely official answer, I would recommend going and talking to your competent authority. Sorry about that, Hubert. I completely forgot to go and mention that. And I will go and disappear off again. Thank you.
Okay, no, thanks, Neil. Now that this is clear, now we can really start. So the outline you see here, we start with the EU regulatory framework for nanomaterials, then discuss the importance of the definitions of nanomaterial, whether they are used in regulations other than REACH, and then we have a closer look on how nanomaterials are covered in REACH. Next slide, please. So when we have a look at the EU regulatory framework for nanomaterials, we see that nanotechnology and nanomaterials, that can be found in many applications with the different regulatory context. But there is no specific legislation for nanotechnology or nanomaterials. So there's no such thing as a nanomaterials regulation. Rather, the nanomaterials are either regulated in sector-specific regulations or in horizontal regulations. But in general, we have to realize, or the European Commission realized, that the EU regulatory framework in principle covers the potential risks posed by nanomaterials, and it applies to nanomaterials even if they are not explicitly addressed or even mentioned in regulations. When we look at newer EU regulations, we can see that they explicitly address nanomaterials. For example, the new novel food regulation, the information on the provision of food information to consumers, cosmetics regulation, biocidal products regulation, medical devices regulation, or the latest one, the REACH, with its amended annexes that explicitly address nanomaterials. So REACH is a horizontal regulation. So it covers chemicals, substances, independent of their use, independent of specific sectors, and therefore it's a horizontal kind of overarching regulation. And then we have the sector-specific regulations such as cosmetics, biocides, and so on, that cover specific products, specific sectors, and so that means certain ingredients, chemicals may be covered by REACH as well as sector-specific regulations. But we will see this later. Next slide, please. So I have here a little table, not necessarily exhaustive, on the EU, that shows the EU regulations that explicitly address nanomaterials. And we have here a number of regulations, REACH, biocidal products, and so on, and several things that are how nanomaterials are addressed in these regulations. So some of those regulations contain definitions of nanomaterials, regulatory definitions of nanomaterials, or nanoforms in the case of REACH. Some of those regulations, they imply a specific approval procedure for nanomaterials if they are used in the context of those regulations, and most of them require a nanospecific safety assessment. That means a safety assessment that takes into account the fact that some materials, ingredients are nanomaterials. And then there are some other legal consequences. For example, in some fields there is a labeling obligation, so products that contain nanomaterials need to be labeled in the realm of certain regulations. And for others, there is a specific guidance on how to get a better understanding of how nanomaterials are addressed in specific legislations. So we have quite a number of regulations. And now let's have a look at the next slide. The fact that nanomaterials are addressed in a regulatory framework in legislation, it makes it necessary that we have clear definitions of the terms that are used. And one of those definitions, or the definition that is important for us here, is the definition of nanomaterial, so regulatory definition of nanomaterial. And that definition of nanomaterial was adopted by the European Commission in 2011 upon request by the European Parliament. And this definition is intended to be comprehensive, so it should include a wide variety of potential nanomaterials. It should be broadly applicable across regulatory fields, and important, there should be no connection, no connection to hazard or risk implied by the definition, by the categorization of a material as nanomaterial. It is a recommendation of the European Commission, so that means it is legally not binding. as it is this regulation. But let's have a look at the definition. What are the main elements? So it says nanomaterials means a natural, incidental, or manufactured material. So that means the origin of the material does not matter, and the material should contain particles. So it's particles are the main things to be analyzed. And those particles can be in an unbound state, or as an aggregate, or as an agglomerate. So the particles can be by themselves or in larger aggregates, agglomerates. And if 50% or more of those particles, so this is the number-based size distribution, 50% of the number of the particles have one or more external dimensions in the size range between 1 and 100 nanometer, that material is a nanomaterial. And what is important is the external dimensions of the particles. And then it has a derogation that says that fullerenes, graphene flakes, and single-wall carbon nanotubes, even if they have one or more external dimensions smaller than 1 nanometer, they should be considered as nanomaterials. And then it has an additional criterion besides the particle size. So it says that if it's possible and requested in a specific legislation, the compliance with the definition can also be determined on the basis of the volume-specific surface area of that material. And if that volume-specific surface area is larger than 60 square meter per cubic centimeter, that material is a nanomaterial. So this is the recommendation of the Commission for definition of nanomaterial. Go to the next slide, please. In the case of REACH, this definition is the basis of another term, namely the definition of nanoform. And this has been included in the amended REACH annexes that have been adopted in December 2018, so it's now part of the REACH. And here, what is defined is not nanomaterial, but so-called nanoform. And this definition of nanoform is on the basis of the Commission recommendation on the definition of nanomaterial, and it then continues that a nanoform is a form for natural or manufactured substance containing particles. And it continues very much in the same way, very similar to the definition of nanomaterial. So it's similar, but it's not quite the same. Why is that so? And what are the differences? No, the definition of nanoform doesn't use the term material, but substance. But substance is a term well defined in REACH, so it uses this term rather than material. Then it does not, this definition of nanoform does not include incidental materials or incidental substances. It only relates to natural or manufactured substances. There is no flexibility of the 50% threshold, whereas the general definition recommendation allows for some flexibility in the threshold. If required in specific regulation, the 50% threshold is fixed here in the case of nanoforms. Then this volume-specific surface area criterion is not there in the definition of nanoform. The most important difference is that this is a legally binding definition for in the scope of REACH. So essentially, it has the same size criterion as the EC recommendation, so the size range 1 to 100 nanometer and the 50% of the particles. If a substance then fulfills the definition of nanoform, it includes specific information requirements for nanoforms, but I think that Neil will in the second part elaborate on that further. So if a substance is a nanoform, then the registrant has to provide information on the size distribution, on the aspect ratio, on the surface functionalization, and on the surface area of that substance when it is registered. Then, yeah, this is the next slide. Then we have also a very interesting term called set of similar nanoforms, also in this amended annexes that says a set of similar nanoforms is a group of nanoforms that are characterized in accordance with that section 2.4, and these are the criteria that define a nanoform. But there have to be clearly defined boundaries in those parameters that define the nanoform, so that within the nanoforms within a set defined that way, they allow us to conclude that the hazard assessment, exposure assessment, the risk assessment of these nanoforms can be performed jointly. And this has to be justified by the registrant, how a set of similar nanoforms is defined, but it is a possibility to assess at the same time several nanoforms if they fulfill certain criteria. And I think that a guidance on how to define such sets of similar nanoforms is expected, and I think ECHA is working on that. Next slide, please. So coming back now to the regulations, the European Commission's recommendation on a definition of nanomaterial has been implemented in several pieces, in several regulations, so it has become a regulatory provision that needs to be respected. It is, for example, incorporated in the new medical devices regulation with slight variations. It is incorporated in the Biocidal products regulation, also with slight variations, depending on the application. And as I just explained, it can be found in the amended annexes of REACH, where the definition of nanoform refers to the definition of nanomaterial. In the food sector, you might want to have a look at the new guidance issued by EFSA on risk assessment of nanomaterials in food and feed applications. This refers also to the EC recommendation of nanomaterial. Next slide, please. So this has legal consequences. There is no time to explain these in details, but we will hear something about that a little bit later. Now, how to implement the definitions, how to find out in practice if a material fulfills the definition of nanomaterial. It has been realized soon after the adoption of the definition that some additional work is required, so to let stakeholders know how to implement it, how to find out in practice if a material is nanomaterial. And there was one EU research project, Horizon, an EU research project called NanoDefine, that during 4 years developed approaches on how to support the implementation of the nanomaterial definition in practice. You might want to have a look at the project web page. The project is now over, so the results are there. And you can find a lot of information how to find out in practice, what to do if you would like to know whether material is nanomaterial or not. And then there are new reports by the JRC to support the implementation of the nanomaterial definition. One of them, called an overview of concepts and terms used in the European Commission's definition of nanomaterial, just published on the 13th of February. So you can download it. The link is at the lower part of the slide. You can find information how to interpret terms and concepts used in the definition. And there will be another report on measurements. I call it for the moment the measurements report. So that will contain a lot of information on how to measure materials, what to measure exactly, and how to evaluate those measurements in order to find out whether material is nano or not. So this report is in progress, and it will be published most likely at the end of this year, 2019. Next slide, please. So now let's have a short look on regulations which explicitly address nanomaterials. We start with the Biocidal products regulation. So this regulation uses the EC's recommendation of a definition of nanomaterial with slight modification. So it applies to natural or manufactured active substances or non-active substances, as these are substances that are regulated by the Biocidal Products Regulation. And it has some provisions, some legal provisions. So it says, for example, that if nanomaterials are used in a biocidal product, they must be explicitly approved as nanomaterials. And this involves a risk assessment, and this risk assessment has to be made separately for nanomaterials, even if there is a substance of the same chemical composition, but not fulfilling the definition of nanomaterial. And then if a biocidal product contains a nanomaterial, this must be indicated in the label. So the label must indicate the name of all nanomaterials, followed by the word nano in brackets. And then tests submitted and carried out and submitted for the approval of the product, they must be explicitly appropriate for nanomaterials, and this has to be justified by the registrant. Currently, a list of approved active substances is available from ECHA, so you might want to have a look at the ECHA website, where you find more information on nanomaterials approved for within the scope of the Biocidal Products Regulation. Next slide, please. Then we have the Medical Devices Regulation, also quite new. It has also a definition of nanomaterial, and it uses the EC recommendation. And briefly, what it says that all devices, when they incorporate medical devices, if they incorporate or consist of nanomaterials, they are classified either in class 3, 2B, or 2A, depending on the potential for internal exposure to nanomaterials. So if they're high or medium, they go into class 3. If they have a low potential for internal exposure of nanomaterials, they go to class 2B. If they present a negligible potential, they go to class 2A. Next slide, please. And then, so these were the regulations that use the EC's nanomaterial definition with some variations. Now we have other regulations that came into force before the EC's recommendation of a definition of nanomaterial was adopted, and naturally, and they contain definitions of nanomaterial, but they are not the same, because that definition did not exist when these regulations were being developed. So let's have a look at the Cosmetics Product Regulation. This was the first EU regulation that explicitly had a definition of nanomaterial and explicitly addressed nanomaterials within its scope. And there the definition goes: a nanomaterial means an insoluble or biopersistent and intentionally manufactured material that has one or more dimensions or internal structure on the scale from 1 to 100 nanometer. So it has some elements that are quite similar to the EC's recommendation, but it has other elements that we do not find in the EC recommendation. One of the reasons is, except the historical reason that this Cosmetic Products Regulation was adopted first, that Cosmetics Product Regulation should guarantee the safe use of cosmetic products. Therefore, it has some the nanomaterials definition used there takes into account some risk considerations. So the solubility and the biopersistence are such considerations. These terms need to be, let's say, explained a little bit more. At the moment, it is not 100% clear how they should be interpreted. So maybe it's case by case. But anyway, this definition is legally binding, and it also requires that cosmetic products that contain nanomaterials, that this should be indicated on the label of the product. Next slide, please. One consequence of the Cosmetics Products Regulation is that, there is the obligation for the Commission to produce a catalogue of nanomaterial, to publish a catalogue of nanomaterials used in cosmetic products that placed in the EU market. And this first version of the catalogue was published in 2017, and the entries you find there are based on the information submitted by the registrants to the Cosmetic Product Notification Portal. And it is updated regularly. Currently, it has it contains some nanomaterials, colorants used as UV filters, and in other functions. And this is an informative list, of course, and it's not a list of authorized nanomaterials, not necessarily. And down here you find an excerpt of the UV filters authorized as nanomaterials, and the entries look like this. They have a name, the category of cosmetics where they are used, the exposure route, in that case it's all dermal, and whether it's a rinse off or leave on product. You find the catalog on a specific website that is also mentioned here. Next slide, please. Then in the food sector, we have also a definition of nanomaterial. In that case, the definition of nanomaterial is found in a novel food regulation, and it defines engineered nanomaterials. So engineered nanomaterials are intentionally produced materials that have one or more dimensions on the order of 100 nanometer or less, or consisting of discrete functional parts on the surface or internally, and many of those should have one or more dimensions on the order of 100 nanometer or less. And it includes also structures, agglomerates, aggregates, that may be larger than 100 nanometer, but they retain properties that are characteristic of the nanoscale. And it is explained further down that properties characteristic of the nanoscale include those related to the large specific surface area and other physico-chemical properties that are different from those of the non-nanoform of the same material. And this is valid in the food sector. Next slide, please. Specifically for within the novel food regulation, but it is also used in the context of the FIC regulation, the regulation on the provision of food information to consumers. In the novel food regulation, one has to realize that the novel food is defined as a food that has not been used in the EU to a significant degree before 1997. And nanomaterials, if they are used as ingredients in food, are therefore novel food if they are used after 1997, and the relevant provisions in the novel food regulation apply. For example, specific approval, a safety assessment, and so forth. There is also, independently from the definition of novel food, there's the label obligation in the food information, consumers information. So if a material fulfills the definition of nanomaterial as laid down in the novel food regulation, products that contain nanomaterials need to be labeled. So in the list of ingredients, nanomaterials need to be indicated with the word nano. More on the risk assessment on nanoscience and nanotechnologies in food and feed chain, you will find in the latest EFSA guidance published recently, and you will find the link is down here on that page. It was published 2018. So next slide, please. Then we have also the plastic food contacts material regulation. That does not contain a definition of nanomaterial, but it says that nanomaterials must be explicitly authorized if used as plastic food contact material. And those that are explicitly authorized as plastic food contacts material are listed here. So in February 2019, we have exactly 6 nanomaterials, and the list, to understand this completely, one has to look in that annex where you can find the list, because the restrictions apply, because these materials listed here are specified in greater detail in that list. So it does not apply to any titanium nitride, for example, but to a specific form of nano titanium nitride. These specifications you find in the list. And also an important thing is that if a material is authorized as food contact material, if the non-nanoform is authorized, it does not cover the nanoform. So therefore, a separate safety assessment for the nanoform is required, and an explicit authorization of the nanoform to be used in plastic food contact materials is necessary. And this concludes my part on the general EU regulatory framework of nanomaterials and the definition and around my discussions around the definition of nanomaterial. And I now hand over to Neil to talk about nanomaterials and REACH. Please, Neil.
Thank you, Hubert. Hubert, can you hear me?
Yes, I can hear you.
Brilliant. That means everyone else can hear me. That's good. Okay, so I'm going to give you a little bit more detail on what's included in the update to the annexes of REACH that was put in force December last year. But I thought firstly I'd just give you a brief overview of the requirements of REACH and in particular how they were looking at nanomaterials beforehand. So anyone who's familiar with REACH will know that each registrant, be they a lead registrant or a joint registrant, should provide chemical characterization of their own substance that's in their products. And this is to identify and quantify both the substance and any major impurities therein. So it's been very much a chemical characterization. However, within the guidance published by the European Chemical Agency, they have suggested that other parameters could also be used within the characterization, such as particle size and whether the substance is a nanomaterial or not. However, these were recommendations in the guidance as opposed to obligations within the regulation. But the reason why people were expected to do this characterization is to ensure that the hazard and exposure and risk assessment that was included in the dossier that's submitted is actually applicable to the product they're putting on the market. This is because only one set of data should be provided per substance to avoid excessive animal testing. So the lead registrant submits all the data, and the joint registrant then goes and buys access to refer to it. But clearly they have to show that the data is relevant to what they're placing on the market. So what happened? Now, please, if there's anyone from one of the legal firms in Brussels, yet again, I have massively simplified this and I apologize it, but I think it makes it quite easy to understand. What was happening? Well, during the evaluation process, the European Chemical Agency and the competent authorities, not just titanium dioxide, but some other substances as well, looked at the dossiers that were submitted, and they highlighted that there wasn't really enough information on the different potential nanoforms of the substance in the characterization, and they wanted to have more detail on each of the separate nanoforms. However, industry, the relevant registrants replied saying, well, this is not actually required in the regulation, and this would be giving excessive requirements for particular substances over other substances, which is not really in line with REACH. So it went to the Board of Appeal, and the Board of Appeal largely found in favour of the registrants because no, there weren't these requirements explicitly stated in the regulation. However, they did highlight that they did acknowledge that without this data, it would make it very difficult for the agency and the competent authorities to actually make a full assessment of whether these substances had been assessed properly and actually were being used safely in the market. So how was this issue addressed? Not surprisingly, perhaps, the regulations have been updated to go and be more explicit in the requirements for nanomaterials and, as the terminology now is, nanoforms. And this has come into force in December last year. It was officially published in the official journal of the European Union. However, clearly, because it's potentially giving more obligations to individual registrants, the registrants do need time to actually put these into force. So there's a deadline to actually meet the obligations that were put into force in 2018 and to either update dossiers, or if you're introducing a new product on the market, include it in a registration dossier. And the deadline for this is January the 1st, 2020. So what do you have to do? So we've moved on from the regulatory side, and we're now moving on to the practical side. First thing it needs to do is particle characterization. And this very first point is the one point that I was a little cautious about putting in the presentation, because it's not absolutely explicitly stated within the regulations. However, it can be implied from the regulations, and within formal discussions with the European Chemical Agency, they view that registrants should be actually looking to see if they have nanoforms, as opposed to it only applying to people that are deliberately placing a nanoform on the market. So I suspect that this particular issue is going to be clarified a little bit later on, but in terms of this informal guidance that I've been given, it is something that perhaps people who have already done registrations on powders, particularly very fine powders, really should be looking at actually investigating whether they have a nanomaterial or not, be it on purpose or not. Now, as we'll see in the later slide, how you do this is not really trivial, and I know a lot of people may well do particle measurement using something like DLS, differential light scattering. However, as we'll see, that doesn't necessarily answer the question of whether you have a nanomaterial or not. So I would recommend that anyone who's making powders should at least be aware and keep an eye on the various guidance documents that will be coming out from the European Chemical Agency in the future. As stated previously, if you do find out you have a nanomaterial or a nanoform, then you got to assess, well, do I only have one nanoform, or am I placing multiple nanoforms on the market? Because I said each set of similar nanoforms will require their own individual entry for particle characterization, as well as we'll see later, the hazard and risk assessments. And so what's needed in terms of particle characterization if you have a nanoform? It's size distribution, shape, surface area, and surface functionalization, or the method you used to do the surface functionalization. In terms of to characterize all nanoforms, will it be necessary to characterize all the nanoforms? Well, it's characterizing each similar set of nanoforms. So it's up to the registrants to decide, do each of my products, my different nanoforms, fall within a set of similar nanoforms or not? So it's really going to be up to the registrants to decide what they actually have to do regarding particle characterization of different products. Now, the issues around particle characterization. As Hubert discussed about the definition, there are certain issues around the definition that make it a little bit difficult, or more challenging, should I say, to actually decide, do I have a nanomaterial or not? Firstly, we talked about the constituent particles, potentially the unbound particles or the primary particles, some people might know them. That's the particle size you need to measure. However, a lot of size measurement techniques don't really distinguish between what they're measuring. They just look at a particle as it exists in suspension. This may be either a unbound particle or the agglomerate / aggregate. So one particularly key issue is going to be, how do we actually prepare these samples to ensure, as far as possible, that we have a suspension of unbound particles rather than agglomerates? It's not trivial, but a lot of this is going to be supported by either the JRC reports or the ECHA guidance coming up. Second one is size distribution by number. Yet again, a lot of the techniques usually use actually measure distribution by mass or volume rather than number, and the conversion from mass volume distribution to number can introduce very large errors. It's not a very good mathematical approach. If you decide that I'm going to do size distribution by microscopy to go and get a good statistically significant answer, you are going to have to measure hundreds, possibly even more, of individual particles to actually get a statistically significant size distribution. And anyone who's had to do this will realize this is a very tedious and quite time-consuming issue. However, once again, there are image analysis techniques that are coming into play. I do know there's one on the NanoDefine website. So this is something that definitely you need to go and look at what is the latest information out there. And finally, the issue we're trying to look at is the shortest dimension. So it's a sphere, all the dimensions are the same, that's easy enough. But if we have a needle, say, we're wanting to go and measure the shortest dimension, a lot of specially sized distribution methods actually will measure the longest dimension, because you're looking at the particle as it tumbles through the media. So a couple of tips for how you might be able to do this. You're going to probably have to use multiple methods. You'll have to probably use one method that looks at measuring a distribution. Microscopy is going to be absolutely essential to really validate that approach, and also to be able to tell such things as particle shape. And although it's not part of the nanoform definition, the volume specific surface area is also another good method to go and try and justify your conclusion. So you can potentially refer back to the nanomaterial definition for that. However, it's very key to ensure you use a method that's appropriate to the substance. Example, some microscopy techniques require application of a vacuum to the sample. If you happen to have a volatile coating, you're not necessarily measuring what you're placing on the market. As we said before, these are good guidances from NanoDefine and the JRC that have done a nice publication in Nanoscale Advances that will give you a guidance on this. So we move from characterization to toxicology. First thing is, well, what information do I need to put in? Now the issue around here is, well, we're looking at reach, so there are different endpoint requirements for different tonnages placed on the market, and these are all laid out in annexes 7 through to 10. The way you work out what requirements are needed is the requirements are based on the total tonnage of the substance. So we're not looking at individual nanoforms, you're looking at total tonnage of the substance. So if we look at the example I've gone and written here for one manufacturer who has 4 grades that are placed on the market. Grade 1, the one I use most of, is not a nanomaterial, but we have 2 separate grades that are also placed on the market. You will have to total the tonnage here. So the total tonnage there is going to be just over 100 tons. So for this particular substance, we're looking at the requirements in annexes 7, 8, and 9. And they'll be required for each set of similar nanoforms. And I just actually ought to point out that a nanoform, if it's the same grade, but within experimental error or deviation between different batches, that's allowed. That's still regarded as the same nanoform. That's why I've gone and put this grade 2 made at 2 different sites. They would both be regarded within the same set of similar nanoforms. That's be regarded as the same nanoform. So what's changed within the actual requirements? Physico-chemical endpoints, partition coefficient. Rather than doing a partition coefficient, because it's really not relevant to nanomaterials, you should be using looking at dispersion stability, and there's no ECD guidance for this. If you then want to move on to soil absorption coefficients, then if you choose the method carefully, then you can use the accepted methods already in the REACH guidance for a nanomaterial. The other big issue is water solubility, because another key thing, not just in terms of just pure, is it dissolved or not, the issue of nanomaterials that really defines on how it may well behave in the environment or in the human body is the rate of dissolution. So this is another aspect that should be looked at within the water solubility for nanomaterial. And in certain areas, it also be important, as we'll see later, to actually not only do water solubility in distilled water, but also look at both environmentally relevant media and biologically relevant media. So perhaps synthetic lung fluid, something like that. Toxicological endpoints, what's changed? It's all very much based on science. There's no ridiculous requirements that aren't based in science. Ames test. An Ames test is a test using bacterial cells. Particles don't cross the bacterial cell wall. So if you show a nanomaterial, especially insoluble one, in an Ames test, you're going to get a negative result. And that's not because it's not mutagenic, it's because there's no exposure. So there's a requirement to move and actually go onto the mammalian cell protocols instead. The other issue, particular key issue, is around the route of exposure. It's regarded as most likely route of exposure to nanomaterials will be by inhalation. So inhalation is regarded as the key route. I've already touched upon the biological fluids, and once again, the characterization of the sample as it actually exists in the study may also be useful, particularly if you're looking at doing grouping and read across. Go to ecotoxicological endpoints. I'll go through this very quickly. We've all touched upon that dissolution rate and dispersion stability are key to work, have a good idea of the environmental fate. There are certain waivers that are applicable to what I would call a normal substance, a bulk substance, that are not applicable to nanomaterials, because even if a nanomaterial isn't dissolved, because it's so small, it's still bioavailable and can actually cross biological boundaries. So perhaps long-term studies might be more appropriate. When we're looking at transformation, we're not just looking at chemical transformation, but also looking at changes of the actual nanoform itself. So coatings or removal of coatings, complexation with other environmental species. Looking at bioaccumulation, there is a test guidance that really looks at bioaccumulation of nanomaterials, and there's an issue. A lot of people tend to use what's known as the equilibration partition method to go and predict sediment and soil toxicity from aquatic toxicity. It's not regarded that this is a particularly sound method for nanomaterials, but then that's also the case for all ionizable substances as well. So we're not looking at something that's solely aimed at nanomaterials. What a registrant is going to have to do is which nanoform are they actually going to use for their studies, and potentially should we really actually try and make a decision on which environmental compartment are we looking at. If the nano form goes straight into the sediment and almost has no exposure to the aquatic environment, should we be looking at sediment toxicity rather than aquatic toxicity? I would probably say here, initially follow what REACH tells you, but be aware that if your substance gets evaluated, the competent authority may well ask you to do further environmental studies if they regard that sediment is really the key environmental compartment. We've touched upon hazard, now we're touching upon risk. Quick introduction to how life cycle exposure and risk assessment of a normal substance or bulk substance is done. The substance is made, you'll get exposure to workers, then if it goes into a consumer item, you get exposed to consumers. Throughout all of these particular processes, the substance is released into the environment, and then eventually it'll degrade by either biodegradation or physical degradation. But the hazard is intrinsic to the substance all the way through its life cycle. So it's well, it can be complex, but relatively simple. We look at a nanomaterial. Because we have potentially multiple nanoforms with different levels of toxicity, the potential of the risk assessment of a nanomaterial can be really quite more complex. However, I would definitely note that the whole issue of grouping, of declaring similar sets or sets of similar nanoforms can, what I would call, collapse this complexity down. So in this situation, you may well actually be able to have a good argument that's saying nanoform 3 and nanoform 4 are actually have exactly the same ecotoxicology, so we can collapse them down and only have to do one assessment around the environmental issue. So definitely, and this is something that the grouping projects are looking at, so I'd keep an eye on those. Further issues that may well impact because of these changes. There is a look at, there's actually a proposal ongoing to amend Annex 2 of REACH regarding safety data sheets to require that people actually identify when a substance is actually present as a nanomaterial and actually state which nanoform is present. I'm going to be quite interested to see if this is required, how you actually state this, but that's something that will come about once this process is gone through, and also to include particle characteristics in the section 9 of the safety data sheet. I think the other thing that will be quite interesting will be the fact that there's a potential for the requirement for exposure scenarios that are for nanomaterials might be required, and there may well be extra information provided within the exposure scenarios, but yet we're going to have to really keep an eye open for what's going to actually come about this. I can't make any comment, but for people interested, I would, yes, definitely keep my eye open for this area. Downstream users, we're not just looking at registrants. Because the assessment is meant to take account of the full life cycle, registrants may well want to know whether their customers are actually modifying the product and hence changing the nanoform. However, the customers may well not want to actually give information back to their supplier, because quite often, especially surface modification, is really a a business is very much a confidential piece of business information. So it may well be that downstream users decide that actually if they're supplied an exposure scenario that doesn't cover their nanoform, they may well want to do their own downstream user chemical safety report, or perhaps if they're not making so much, at least do their own assessment. So please note that downstream users may well have additional requirements to do as well as registrants. So potential matters of interest. So I had to go and slightly change this title to being slightly more neutral. So as we've already seen, identification of a nanomaterial is not necessarily straightforward. And our own personal experience is you can send the same sample to 3 different labs and get 3 different results. So this is probably due to sample preparation. So I'd say if you're going to use a lab, make sure you're choosing a good one and ensure that they are really clued up on all the guidance documents for sample preparation. Second thing is going to be, how are we going to set the limits for a similar set of nanomaterials? What is scientifically justified actually going to mean? Guidance is on its way, but when it comes down to it, it's going to be up to the registrants to justify this, and then if the substance is ever evaluated, the evaluators will decide on whether they agree with the justification or not. If you try and do all the testing for all the different similar sets of nanoforms, or sets of similar nanoforms, should I say, if you do the full testing, this will be very expensive and do really quite a lot of require a lot of animals. We don't want to do this, so the issue around grouping and read across is going to be absolutely essential, and I'll cover that on the next slides. Validated protocols available for all endpoints. There are lots of new guidances becoming available under development, so I would definitely keep an eye out for things. And thanks to Hubert, he sent me this link, which really gives the absolute up-to-date situation regarding OECD test guidelines. It has been accepted by the journal, and he's expecting it to be published within the next couple of weeks. So I would definitely keep your eye open for this in terms of what protocols are available. I think a very key issue that people are going to have to be very keen on is, how are the various joint registrants of the same substance actually going to communicate with each other regarding setting up sets of similar nanoforms, or trying to go and set up a read across approach? Because as I said before, quite often the particle characteristics are intellectual property of a company, and really they might not want to go and communicate this to other competitors. It's up to the registrants. Yet again, the regulators are sort of saying, no, it's up to you to go and decide how you're going to do it. So I would definitely say that joint registrants, you may well have to talk to each other again, even though you think you finished all your registrations, just to go and decide how you're going to do this to ensure that you maintain your intellectual property. So we're going to move on to grouping and read across. So grouping and read across is used in REACH already. It's an alternative to testing to try and reduce the amount of animal testing. And it's where you try and use existing data on a data-rich substance to fill in the gaps with a data-poor substance. So, and say, there's lots of guidance out there already for all the other substances on REACH, and there's guidance also for nanomaterials. Obviously, because we're now looking at different sets of similar nanoforms, we're probably going to have to look at, well, can we do read across and grouping for a nanomaterial across its different similar sets? So between nanoforms, potentially we'll look to go and see, well, we've got lots of data on the bulk. Can we actually use the same data on the nano? Can we justify it? And Gracious, as I'll introduce on the next couple of slides, is really looking at trying to come up with methods to do this. I would state that also these grouping methods will also probably be needed to actually justify the setting the boundaries of the set of similar nanoforms in the first place. So not only are we looking using grouping to go between sets of similar nanoforms, but we'll probably also need it to actually justify the actual setting the boundaries of that set of similar nanoforms. So what's research on this? I'm going to highlight Gracious, but I'd also like to highlight another national project called Nanograveur, which is also looking at this. And I know there are other ones as well, but I can't fit them all on the slide. So Gracious is looking to develop a framework and guidance document supported by case studies to allow grouping and read across of nanomaterials. And the way it's going to do this is to guide people on the identification of hypotheses for the grouping, and then to go and guide people on designing good experiments to actually test them, with a priority clearly being given to non in vivo, so that means non-animal testing studies, so be that in silico or in vitro tests. Clearly the justification is going to be very, very important. So toxicokinetic and environmental behaviors, such things as mode of action, are going to be very, very key to actually justify these potential groups. And just quickly, my last slide, examples of some of the hypotheses that are being looked at, and perhaps the questions that a registrant could think about within the CEF. One hypothesis: high aspect ratio nanomaterials, such as needles and plates, have chronic inhalation concerns. That's sort of really it's well known around that asbestos is a high aspect ratio material, not necessarily a nanomaterial, but it is a high aspect ratio. And so that's the, I would say here, what aspect ratio actually defines a high aspect ratio nanomaterial. There is a 3:1 guidance from the World Health Organization, but it really is regarding asbestos, where greater than 3:1 is a high aspect ratio and therefore has chronic inhalation concerns. Does the 3:1 ratio apply to the nanomaterials or different substances? That's something that you would have to go and investigate within the grouping experiments. Does the solubility affect it? If it's very soluble in biological fluids, does it really matter whether it's a high aspect ratio or not? And what is the solubility that means either it is hazardous or not? On example 2, another potential hypothesis is that rapidly soluble nanomaterials can be treated the same as the bulk forms, because they all dissolve to give the ionic form. Questions you might want to ask is, well, what rate of dissolution actually is regarded as rapidly soluble? So these are just questions you want to ask. They are experiments that are being done by the Gracious team members, so they'll appear in the Gracious website, so you'll be able to keep an eye on how these are going if you keep an eye on the web page. Thank you very much for your attention.